Hi, this is part 7 of Neurons 2. So what we get to here is we've talked about the propagation of an action potential. Now how does it pass that signal on to the following cell? So we have a presynaptic cell in this diagram here and a postsynaptic neuron. So the presynaptic cell is going to transmit its action potential and deliver it here through the axon terminal to the next cell, which is the postsynaptic cell. And so what we want to look at is that little synapse, that space where they connect. This is a reminder slide talking about synapses. They may be chemical or they may be electrical, but remember that the electrical ones are rare. Those are the gap junctions in the heart or neurons in the central nervous system. We also have some that are in the pulp of your tooth and in the eye. So we sort of pinpoint out where these are. And as I said, cardiac and also smooth muscle in the smooth muscle of the digestive tract. Still, not as common. This kind of synapse is very quick because you don't need to release a neurotransmitter, and that action potential is going to jump from one cell to the next. So it is very fast, and you need gap junctions for this to happen so that you have that connection between both cells. It's going to allow that depolarization to travel from one cell to the next. Most common, however, are chemical synapses. This is most of them, and they will use a neurotransmitter in order to send that message to the next cell. This provides one-way communication, correct? It's going to only go one way, and we're going to look at that anatomy of a chemical synapse. In this diagram, there's lots of vocabulary, and I've listed that for you. So we have a presynaptic membrane. So this cell with its membrane is the presynaptic membrane. The postsynaptic membrane. So this is cell number 2 down below, and that would be the postsynaptic membrane, the membrane of the cell receiving that signal. Between those two, there's going to be a synaptic cleft. So that space in between here is the synaptic cleft. Let me get my pen going. Continuing along, the synaptic vesicles then are going to be in this terminal portion, in the axon terminal here of the presynaptic cell. And all of these are little balls with neurotransmitter in them. They contain the neurotransmitter, and the receptor sites are going to be the ones that receive that neurotransmitter. So they're going to be on the postsynaptic cell here, ready to receive that neurotransmitter and then cause a change in the membrane permeability to sodium once that neurotransmitter arrives at that receptor. So that's the list of vocab that would be on the chemical synapse. Neurotransmitters, a little more on these. They're chemicals released by the presynaptic neuron, and there's many different kinds that we'll look at later on. They're going to bind to the ion channel on the postsynaptic neuron, and they're going to trigger the opening of the channel to produce a graded potential. Remember, this is going to be a graded potential because maybe there isn't enough stimulus, and if not enough stimulus comes here, that graded potential may not be sufficient to reach the axon hillock of this second cell, of the postsynaptic cell. If we were to take it in steps and show how a chemical synapse works, we have 5 different steps here, and I want to show them to you one at a time. So step 1, the action potential depolarizes the axon terminal. So in red, you have an action potential that's coming down along the surface here of this neuron, and it reaches then this axon terminal part, that section. So that's step 1. What happens next? That depolarization opens voltage-gated calcium channels, and calcium enters the cell. Where was the calcium? It's here in this area, outside of the cells, extracellular fluid, and this action potential is going to now open that gate, and calcium has come into the cell. Let's call that step 2. Step 3, that calcium entry triggers exocytosis of the synaptic vesicle contents. That should be familiar because we talked about this before. So calcium now is going to allow for this stashed away neurotransmitter to actually come over to the docking protein and create an exocytosis. In other words, here it is, pump out that neurotransmitter. We talked about this with insulin, remember? 4 would be the neurotransmitter diffuses across that synaptic cleft and binds with the receptor. There's the receptor on the postsynaptic cell. What's going to happen next is that neurotransmitter initiates a cell response in the next postsynaptic cell. So if that neurotransmitter has reached the receptor, then it's going to cause a response in that cell by opening sodium channels that are going to now allow sodium to come in, and we begin this whole action potential is going to start now on this second cell. Correct? You'll have a second one now on that cell. If I could put in a step 6, it would be acetylcholine has to be broken down eventually if you want this to stop, if you want to stop this connection between both cells. So acetylcholine, which is one of the neurotransmitters, and it's stimulatory most of the time, it will break it down into acetic acid, that's the acetyl part, and choline. And so often you have a little diagram where it sort of breaks it in two, and one of them is the acetic acid and the other one is the choline. And by breaking it down into 2 parts, it stops being active and then it can be recycled and used again. But that would be the last step there, where you actually want to stop that communication between those 2 cells. And speaking of stop, this is where we stop this video, and you have one more in this module. Thanks.